Is that fair enough? So today, let me tell you what I'm hoping. Let me tell you what we're hoping to kind of get through today. I do want to introduce this article, and I want to make sure that we look at a couple of things and be, conversant on what this is actually talking about a little bit, right? And then I want to remind you that Wednesday, you do have your first multiple choice test in this class, and this class is very different from for those of you who know me in other classes. And that the only thing that I'll write questions about are what we have actually talked about in here. There's and that I have given you and that we've talked about, right? So today I'd like to make sure that everybody in the room is really comfortable about what notes you should be studying for Wednesday, okay? And so with that in mind, we're also going to spend a few minutes going over your list of microorganisms so you know which ones would be fair for us to mention. So we'll just kind of go down the list real quickly, making sure you have a little check beside the ones that you've been introduced to at this point. The ones that you have not, we're going to skip right over, right? And so fine. Any questions about that? All right. But before we kind of start today with some of the information that we know might be on the thing for Wednesday and covering what might be there for Wednesday, I did want to bring this article to your attention from the Mayo Clinic. And you know how people love to say, I told you so, right? Do y'all know people who are like that? Okay, well, I'm going to be like that for a minute. I wish I had some of my micro people from years ago, because they would be telling you all, I told them so. You guys, we know what a microbiome is, right? But the microbiome means all those microorganisms that are your gardens, where you're allowing you to live and to be healthy. And understanding the balance of the microbiome is just key to us going forward and understanding some of these disorders that we're beginning to see a lot of. So you guys saw that in here a couple of things that were mentioned that you already know about, like the hygiene hypothesis. Did you see that? Which means what? What is the hygiene? Did y'all see that in here, the hygiene hypothesis? You saw that? What does it mean? Yeah, so actually we realized that especially the microbes that are in our gut, our colons, we know that they play a huge integral role now in our immune system, educating our immune system. So which microbes we have there are going to be really important for our immune systems to be functioning optimally. So a couple of things just were really interesting, I think, in here. There's a microbe that is not on your list, but if I were you all right now, I would maybe at the bottom of the bacteria, because we're going to introduce this. And sorry, but here we go. Here's another one. Here's another one. There'll be very, very few that we ever add to this. But at the end of your bacteria, which is blue, right? The blue sheets of paper. So in the next one, let's go ahead and put Protozoa histicola. So this is a bacterium that they know now is definitely maybe a keystone species that you need for your colon to be healthy, right? And all of these autoimmune diseases like multiple sclerosis and rheumatoid arthritis and Crohn's disease and these autoimmune diseases that we're seeing higher and higher numbers of, we're thinking it really has a lot to do with how our microbiomes are not healthy. And figuring that out, hopefully a lot more research is still ongoing right now, but more research is actually happening to hopefully use this as a way to treat. Because could we try to help people reestablish some of these microbes in our environment? You know, a lot of times, they say, you are what you eat and da-da-da-da-da, but we should eat like fresh vegetables and fruit. Well, what has a lot of things on them, fresh vegetables and fruit? What do they have a lot on them? Microbes. So you're reestablishing them and constantly giving that reestablishment, that normal flora. So if somebody's way out of balance, it can take a long time, right? So maybe there would be a way to figure out exactly how to use these to help you treat. I love the little analogy too. So we added a bacteria, didn't we?
Okay, so what do you want to write on the right?
Oh, so so it's not a disease, but what I want you to just put part of normal flora here. We just want to be aware of it because it's just part of normal flora, right? So yeah, there's not really a disease, so that's kind of NA as far as that goes. I love the idea of the idle hands lead to devil's work. So maybe stimulating that immune system is going to be important. Now the rest of this and how it's being investigated in obesity, cancer, inflammatory bowel disease, and C. diff infections. I just think we need to have heard about this. So the rest of it is just FYI, okay? I hope you all will find this interesting. I really did. I find it very interesting. But anyway, do you want to, before we start, do you want to go over our list and see which ones would maybe be fair for Wednesday? And then there are a few little things I want to tie up as far as the PowerPoints and lecture goes, okay? And maybe even just a really quick kind of review for Wednesday. Fair enough? And if I run out, y'all will know why. Almonds. It was probably just that they were the healthiest things I'd eaten in a long time. My body was like, healthy food? Shocking. Okay. So, Staph aureus, is that fair? Yes. Staph aureus. Streptococcus pyogenes. Y'all help me out here. What's the next? Okay, but what's the next one you all have gotten to? So you have Streptococcus pneumoniae. Fair? Fair? Okay. Did we have Agalactiae? No. That was A and B. So don't put that in here. Don't put that in here then. Clostridium perfringens. Clostridium difficile. Y'all are just putting a little pencil mark, aren't you, beside these? Okay. Clostridium tetani. Clostridium botulinum. Bacillus anthracis. Right? Escherichia coli. Right? Now, look. When y'all are putting little checks beside these, there's some things in the diseases that you might not have in your notes, but I will promise you, because you know if I give you a question that we haven't talked about, y'all will let me know. Just know the diseases we've mentioned, okay? But we'll continue to talk about them. Help me out on this next, on the back page. Listeria monocytogenes. Mycoplasma pneumoniae, that's fair. Pseudomonas aeruginosa. Help me out. What's the next one? No more on this one? Okay. This next page, thank you, Pearson. The next page, Haemophilus influenzae. Right. Haemophilus influenzae. And that's a bacterium, not to be confused with influenza virus, right? Not to be confused with that this is a bacterium. We're still a bacterium. How about Neisseria gonorrhoeae? Yep. How about Lactobacillus acidophilus? Okay. And see, and actually, this is another one where we don't have a disease, do we? We said that this is part of normal flora, and it's important that it be the predominant one. Okay. Mycobacterium tuberculosis, is that right? And other species of Mycobacterium. Good. How about Mycobacterium leprae? Okay. How about any more on that page? No? Okay. All right, so we're good. Are we good with bacteria? Gardnerella vaginalis?
No, we did the vaginal.
We did Trichomonas vaginalis, which is a protozoa. Somebody said we did. Did we talk about Streptococcus pneumoniae?
I'm pretty sure we did.
All right, I might have said that. What you said? Okay, so let's just don't put it on this one. Okay, so we just won't put it on this one. Y'all can mark your little pencil mark out for that one. That's fine.
Yeah, that's what I.
It is middle. Yeah, but it's also Streptococcus pneumoniae can do the same thing, and conjunctivitis. But if we hadn't talked about it, it's fine. Okay, so let's what you say, Jay. Was it nice enough to repeat? So it was not. Michelle, was it? Okay, Michelle. Okay. Protozoa. All right, let's look at protozoa. We have maybe spoken of Trichomonas vaginalis. Is that right?
Yeah.
And did I not mention the Plasmodium species that cause malaria? I didn't say that. Okay, well that's fine. That's fine then. Okay, so we're moving along. An AP. Okay. So fungi. On the fungi, we have mentioned Candida albicans, haven't we? And maybe we have also mentioned about Cryptococcus neoformis. Is that right? Okay, we have some ways to go. Helminths. We're in helminths, and maybe I haven't talked about any helminths yet, have I? No, they're in the animal kingdom. You know, they're like little critters, aren't they? All right, viruses. We have talked about measles because of the outbreak. Did anybody go on that site and look at did you go on that and see where there's 80% and then there's 90+% and how quickly the cases spread and how that is? It's really an interesting site. So, okay, so measles we've talked about. Which other virus have we talked about?
Rabies, but that was like something you brought up.
Okay, but we talked, rabies is a disease that's caused from a virus, so that's good to know. Most, the viruses, let's take a look at these viruses. Yeah, sometimes they have these family names that are names you never hear about, but the beautiful thing about viruses names is usually the name is the disease. You know what I mean? So they're kind of easier, aren't they? Right? So any of the other viruses that we mentioned? We've mentioned HIV. So that's on here, isn't it? That you know exactly what it stands for and what it can cause, AIDS-related complex and acquired immunodeficiency syndrome, which is on the next page. Any other viruses?
Kind of influenza virus.
Yeah, where is that? Is that on here? Okay, it's on the next page. Okay, so the influenza virus, good.
Polio virus too.
And the polio virus, great. And we talked about smallpox, didn't we?
Yeah.
Variola, smallpox is eradicated, isn't it? Variola is smallpox, and the smallpox virus, and it's eradicated.
Did we talk about chickenpox?
Oh my
No.
goodness, and did we?
No.
I don't remember saying varicose.
Okay, well, so we won't put that one this time. That's fine.
So which one do we have off this list?
Okay, so off this list, we have measles. We have variola virus, which is smallpox. It's been eradicated since the late 70s.
Rabies.
We have rabies,
Polio.
and HIV, and influenza on the next page, on the other side. No, maybe not. I don't know. That's fine. So that's enough for this one. That's enough for this little challenge Wednesday. And I want to add something for you now because I'm going to talk about it right now. So on this. So on this last page, you see prions and then you see some vectors. Under prions, I'm going to want you all to know that if you thought of any disease or disorder as being kind of scary, right? This one's really scary. Prion diseases are really scary. Prion diseases are diseases that are caused from self-replicating proteins. And I do want you to know about this for Wednesday. So these are diseases that are caused from self-replicating proteins. Now, here's what I need you all to tell me. What do you know about protein production? What do you know about proteins? Proteins are made out. The proteins are the monomers, building blocks for amino acids that come together, and amino acids get in line at a particular time, dictated by what? By DNA. So everything that biologists, including microbiologists, have learned about life, I mean, it is really the cornerstone of life is that we know that nucleic acids, DNA, dictate protein production. And that proteins recipes come from DNA. And that proteins don't self-replicate, right? And that proteins have a shelf life. And that cells are essentially protein factories. Isn't that right? And that protein shapes and structures are dictated by these hydrogen bonds, which are weak kind of bonds. And we can denature proteins. We can play with heat. We can play with pH. We can play with ions like hydrogen ions and change protein structure and lose protein structure. But anyway, our cells are protein factories. So that's been really like a major thing in biology and understanding life, cellular life, until we realize that there are these proteins. Thank God, they're rare. And the stuff of nightmares these proteins are. These self-replicating proteins that if invade your cells, will take over your DNA, cellular machinery, and start making you produce these variant proteins. The scary thing is that they're going to invade central nervous system tissue. And these self-replicating proteins are a progressive terminal diagnosis. What does that mean?
You're dying.
What did you say, Jen?
You're dying.
You're dying. Yes, you're going to die from these. Is there any treatment? No. No treatment. Prions are the things of nightmares. Yes. Which part was it?
Progressive.
A progressive terminal disease. Progressive means that you're going to continue to get worse. Terminal means until you die. And it will be what kills you as long as something else doesn't kill you first. But it will be what kills you. So progressive terminal diseases like emphysema is a progressive terminal diagnosis. If you get diagnosed with emphysema, it is a progressive terminal disease. There's no turning back. You mean you might have a heart attack first. But if you don't, the disease will kill you. And it's going to progress and there's no turning it back. So there's some diseases that are like that. This is an infectious disease, which means what? It means you can what? I want you to know the term infectious and communicable means kind of the same things, is that you can get them from someone. So what this disease is, are you ready for this? It's on your list though. Spongiform, it causes spongiform encephalopathies or spongiform encephalitis. Same thing. Spongiform. Now, you know what that really means? Because, we say there's no such thing as big words. They're just, it means it's going to turn your brain into sponge, holy looking sponges. It's literally going to macerate, chew up, and cause your brain to do that. None of this sounds fun, does it? So, prion diseases. In humans, this disease is called CJD. It's pronounced Creutzfeldt-Jakob. Jakob, it looks like Jacob, but it's Jakob syndrome or disease, CHD. So that's the human variant of this prion disease. And it is thought that people encounter these prions from eating contaminated meat products from something that had the prion infection. We know that in cows, it's called mad cow disease, right? In sheep, it's sheep scrappy or I don't know how you would say that, but in sheep, it's that. And this is why you should be very careful about the meats that you're eating, that you kind of observe the creature before you eat the meat. But not only that, you should also never eat any meat that's contaminated with cerebrospinal fluid or the central nervous system tissues of the animal.
I mean, you wouldn't really know that.
Well, you wouldn't know it from buying it at Food Lion.
Yeah.
except that we do have like USDA, you know, we have investigators as long as the government's working.
Oh, yes.
We have investigators that are actually do, you know, have certain standards and meet standards and whatever. And in developed countries, we do, this is why we don't see this so frequently. So any questions about what a prion is, a prion disease? So this is,
Is that one that you named the only one?
what you just said?
Is the one that you named the only one?
Well, the human variant, yes, the human variant. And then did I put down the animals ones? Yeah, yeah. There was. I want to say it was. I cannot believe I can't think of this. Something like that. But anyway, this happened a long time ago. But there was a tribal society in South America, and they were noting that the young men and older men were dying of this neurologically progressive disease that was devastating to them. But the women were not being affected. So when they went in, and medical anthropologists also went in to try to figure out, epidemiologists went in to try to figure out what was going on. And this horrific, as you can imagine, disease was taking these people out. But it was the young boys and the men. And what they realized is part of their cultural practice was that when a loved one died, the men would actually eat the brains of the loved one that would keep them alive, right? Molly's face. Molly's like, so, but if you think about that, I could see where that would be a cultural practice. I mean, I think that seems a lot more sane than embalming. Embalming is brutal, is just barbaric, what we do to bodies in our culture. So anyway, I think that's, I didn't say I'd do it, but I'm just saying, I can see how that would start, okay, in that practice. What'd you say?
Brain.
Well, how many of you have grown up on farms where people ate the pork brains? You know, when yeah, absolutely, right? So absolutely. I don't eat it that much.
Yeah.
You watch someone else eat it?
Yeah.
Yeah. My dad had mountain oysters and pork brains. Oh man, they were his favorite part, right? And scrambled eggs. Okay, so they're not oysters. They're not oysters. So okay, but anyway, so just to let you guys know that, so they stopped that practice. Was that an easy thing to do, or was that really tough to change people's minds about cultural practices? I mean, that would be really hard, wouldn't it? If you felt like your loved one wouldn't go to the next, you know, whatever, or and if you felt that, I mean, would you find that to be really, really hard? There are people that you all know that feel like if they're not buried in the ground, if they are cremated, that they won't get to wherever they're going, right? Do y'all know people that, right? And so is it easy to change their minds? I bet you've known a lot that won't change their mind. They want to be buried in a steel box, you know, and so it's just really hard to change people's minds. But they ended up going in and doing that. So anyway, prion disease is scary. All right, so are we feeling comfortable about this? If I were to ask you a question that we did not cover on this today, on Wednesday, you all will let me know, won't you? Yeah, you will. So, all right, so I just want to make sure I cover the rest, just a few little things in case I hadn't talked about them. We said eukaryotes. We know that eukaryotes are complex, aren't they? Yes. And by the way, so I think somebody wrote on one of their study guides at Proteins, and it doesn't matter doing it, it's no big deal. Their study guides are really for y'all, and they're really more of a participation grade. I'm not correcting them, but I haven't seen some errors. So protists are actually eukaryotic. They're complex. We saw how big they were the other day, didn't we? We saw some of them are like monster-looking things, right? In the microbial world. But, you know, they're really complicated. They have a lot of stuff going on, these protists, but they are unicellular. So they are single cell, the protists are. And so we understand that, and we understand some of these structures. What the Golgi does, what does Golgi do? Amanda, you good? Okay, what is the Golgi doing? It's wrapping, it's shipping. We know that the nucleus houses the cellular DNA. The endoplasmic reticulum is where proteins and lipids are built. Ribosomes are the workbenches. And eukaryotic ribosomes are really different than prokaryotic bacterial ribosomes. Why is that important to know? Because we can target the bacterial ribosomes with medications that won't be harmful to our own, right? And so that's something we can use. And so great, and mitochondria and chloroplasts, we said, have something in common. Mitochondria is the powerhouse of a cell. Chloroplasts are where photosynthesis occurs in cells that have chloroplasts. Of course, animal cells don't have chloroplasts. But anyway, what's the thing they have in common? They have their own DNA. And because they have their own DNA, we understand that once upon a time, which sounds like a fairy tale, the beginning of a fairy tale, you know, but whatever, it's likely that they were independent, they were living independently as single-celled bacteria, you know, simple prokaryotes. And then got with other simple prokaryotes until now they're in this symbiotic relationship. And it allows for that cell to have way more complex activity, right? The beginning of eukaryotes. And what was that theory? The theory of endosymbiosis, which is kind of explaining how complex life kind of evolved. And so we talked about that. In this article, did you see the dysbiosis that they talked about, where microbes are not working together? Did you see that? Where they mentioned that? Okay, whatever. What do cilia do? If they're projections from the outer membrane that can help, if they're single-celled organisms, can help the cells to move. And multi-celled organisms, they have very different functions. They're usually moving things past the cell. But in single-celled organisms, they can help it to move. And anyway, so that's that. The glycocalyx was what I call your sugar coating. And so the glycocalyx is that layer that has all of the self-identity markers. And those are antigens that are going to be unique to that particular organism, right? So do y'all know about this one? Abigail, you're looking at me like I don't remember. Oh, we did not go over this. This was an A&P. We did. But if we didn't in here, do y'all want me to tell you about it for a minute? Yes. Let's go over this for a minute. Because what this really is that it is on the outermost layer, and this is going to be the layer where you find those antigens, which are proteins, that are unique to each particular species. So they are so unique, I talk about them as being the outfits for the particular species. So for example, they're so unique to each particular species, it would be as distinctive for you is if you were to see, I talk about Elvis, you know, when I say the term Elvis, is that dating me? Elvis? I love Elvis. Thank you. Me too. So much. Okay, so anyway, Elvis. If Elvis were to walk in here, we'd expect him to look a certain way, right? And we would know him from just how he's looking, right? But would he have to walk in here face forward, or could he walk in here backwards and we'd still know him, right? Because he's unique, right? It's his outfit. It's his sugar coating. We each have one. We each have one that is unique to us individually, right? And within a species, they have they have similar ones too. So this outfit is found in these antigens, they're called antigenic determinants. But anyway, these antigens are what you just call the outfits. And they're found here in this particular layer. Did we talk about a capsule and how if an organism has a capsule, that can actually help that organism to be more virulent? And we'll talk more about that in a little bit. I mean, not today, but in another time. Okay. So now when we think about cell walls, do animals have cell walls? No. Animals don't have cell walls. Prokaryotes, most of them, not all, but most of them do have cell walls. And we know that fungi and plants, and other kingdoms, they have cell walls, but animals do not, do we? So, okay, and that's pretty much kind of all I want to talk about with that, the cell walls. We know that even in the bacteria that do have cell walls, the gram-positive bacteria cell walls are very different from the gram-negative bacteria and their cell walls. So, you know, can we use that when we're trying to treat?
Yes.
Think about treating them. Yeah. All right. I had talked about the cytoplasmic. I'm just kind of going through. Y'all know what a nucleus does. We knew what the endoplasmic reticulum and the Golgi, mitochondria, chloroplasts, ribosomes are the workbenches. Is that right? Cytoskeleton. This is this protein highway system. Cytoskeletons are this protein highway system. Highway systems that are going to. They're going to be this protein kind of highway systems where inside of cells, and we have cytoskeletons too, but this is how things are transported throughout the cells. And so we understand that this transport of moving substances within the cells are important. I would have you just kind of look and notice that there's a chart in your book called just the basic comparisons of cells versus viruses. And we said viruses aren't alive. They're considered to be active or inactive. Is that right? Mm-hmm. So we understand that they just don't have a lot of these things that we were just talking about as far as life processes. So this is just something that you could look at. We talked about fission, prokaryotic fission, as being how bacteria divide, which is a type of cloning, isn't it? Right. Now, fungi, just some basic things about fungi. We know that's in the eukaryotic domain. It's a kingdom all its own. There are some fungi that are single cell, are called unicellular. These are yeast. Yeast. Y'all have heard of yeast, haven't you? Mm-hmm. Completely different than bacteria. Yeast are in fungi kingdom, not at all like bacteria, really. Way more complicated than bacteria, okay? Some of them not as complicated as others, but definitely way more complicated than bacteria. Some can be unicellular, some can be multicellular, like mushrooms. Who loves mushrooms? I love mushrooms, okay. Maybe not after I eat almonds, but I like mushrooms usually when I'm thinking about them. Okay, so anyway, yeah, moles. So moles, these are just a special kind of the multicellular ones. You can Google them. They're actually really beautiful. They really do look like puffballs. That's what they look like. So anyway, and whatever. So these you know are in the fungi kingdom. Now, some basic things about fungi. And mold is micro or macro? Mold is macroscopic. Well, you know, you can see molds. You don't always see every bit of molds because molds, these produce spores, and spores are microscopic. And you can inhale spores, can't you? And so you could do that. But when we think about molds, has everybody seen mold on bread? They have little hair-like little, you know, molds, cheese, jelly. Y'all are acting like, what does her refrigerator look like? But actually, you can see mold. Mold is actually yeast, fungi, I should say. These are called saprobes, and S-A-P-R-O-B-E, saprobes. And they can live in really kind of harsh environments. They really can. So, you know, they'll grow anywhere. How many of you all have been up in attics or cellars or whatever? You can see them on old newspapers, photographs. Has everybody seen that? Books, right? You can smell. Sometimes you can smell it, can't you? No? Okay. What'd you say? Already then. So anyway, yeah. So were you asking about the macroscopic because you're wondering why we talk about them in micro? Sure. Is that why you asked the question? Yeah. Okay. Because actually it's a good thought process, but it's oftentimes because the spores are microscopic, but also yeast are going to be, unless they're growing on culture, you're not going to be able to see them, right? And we have these on our skin. Now, you do not have to add this to your list now. You don't have to. But we have, what you said?
I'm just saying that means we have to.
No, you don't. Don't add it.
To write it in our notes.
No, because it won't be on the test Wednesday. So just don't add. It's on your list. But you know, we have Candida albicans as part of our normal flora, don't we? And depending on what's going on with us, we might have more than we should, and that gives us what we call yeast infections. Remember Candida albicans? We had talked about that. So if you've ever seen somebody with diaper rash, that's Candida albicans. Diaper rash is from Candida albicans. Have you seen it? Almost looks like scalded skin, doesn't it? You all seen that? Diaper rash. Thrush. Who's seen thrush before?
What is that?
Thrush they're going to be these little white patches that you can see, and oftentimes babies' mouths. But that's because babies have kind of an immature immune system, and their normal flora is not quite balanced yet. And so a lot of it that can be off. So you can see it there. You can see it on patients, older patients sometimes that have whose immune systems are compromised, that have maybe been on chemotherapeutics for a long time, and the yeast start taking over. So you'll see these white plaque patches in their mouth, and it's called thrush. So diaper rash, thrush, yeast vaginosis, a vaginal yeast infection, vaginitis situation. So we have Candida albicans, but y'all have probably also heard of ringworm. And just for your notes now, but I won't ask you this for the test, Lindsay, but for your notes now, so you can go back later and at it. Ringworm, you've all heard of. Has anybody ever seen ringworm? You have? And when you see ringworm, it causes almost this worm-like appearance in the skin. It can be in the scalp, and it can be anywhere on the skin. It was actually called that because it can really look sort of like there's a worm, you know, whatever. But anyway, it's actually fungal. It's a fungal infection. How many of you seen people who look like when they're tanning, they'll get these white patches that almost look like round little, that are losing color. So they'll be tan, and then they'll have these areas where the color is lost. That's Malassezia furfura. That's a yeast that's on everybody's skin. But if it's overgrowing on the skin, it can actually cause that appearance. It's called tanning versicolor. So, you know, we have fungi around.
Can you explain saprobes?
Oh, I probably never finished it, did I?
No.
Thank you very much, Sarah. So, okay, so a saprobe, they are saprophytic, which is really important to know about what they do. Because the whole kingdom is this way. They live off of dead material. That's what a saprobe does. Yeast actually secrete a digestive enzyme. This is the way that all fungi do. They secrete or excrete a digestive enzyme for dead organic matter that dissolves the matter, and then they suck it in. So they're decomposers. Fungi are decomposers. If we did not have the fungi that we have on our planet and the balance that we have on our planet, we would be so deep in organic trash that we wouldn't survive. We have to have fungi and at the rate we have it, because they're decomposing and putting back into our environment those monomers, those building block units, at the rate that we can do it, so that we can all use them again. Living organisms can reuse them again. So we're glad of decomposers, aren't we? And they're saprobes. They live off of dead organic matter. Okay. So the next time you say, ooh, a fungi or mold or whatever, you kind of don't want to do that, do you? Now, can they cause problems? Yes. Well, so yes, they can. So there are going to be some that are pathogenic. We're going to talk about some that are going to be pathogenic, but we won't add them to our list today. I just wanted you to know about some of them, okay? Do we use them for food products?
Yes.
Yeah, of course we do. Do we use them for antibiotics? Yeah, which one comes to mind? Penicillin, right? So penicillin, it definitely comes to mind. And so we know that we get penicillin from Penicillium the mold that's called penicillium. So, okay, we know that maybe they cause problems for some of our food sources. So does this actually increase like our, the cost of some of our food if we know that we're going to be losing a lot of it to this? Yeah, and so we do know that, and it can actually cause, there can be harmful things to it, but there's certainly lots of benefits as well. Here's the saprobic properties or saprobe, as you said. So anyway, what else? I don't need you to see that. We're going to see some of that in lab. We do know that if they can have asexual or sexual reproduction, they can have one or the other. So asexual or sexual reproduction. Pardon?
What can have that?
Fungi. Fungi can be sexually or asexually reproducing. We think about these spores that they can give off. And that's kind of all that. I had already talked to you about protozoa, and we're going to add to our list of protozoa. We know that they're single celled, aren't they? So these are actually single celled. This kingdom, Proteus, is kind of a kingdom that everything gets put into until we know better. And then sometimes when we figure out at a molecular, what the RNA and whatever looks like, then we can actually place it into another kingdom. But this is kind of the catch-all kingdom the proteins are at first. The trichomonas vaginalis is the only one that I've given you so far. And we said that people call this a trick infection, don't they? This little protozoa is just like the ones you all were seeing in lab the other day. It's not as big. They're not as big as the paramecium, but they're way bigger than the euglena. So if that gives you sort of an idea about how big they are, and they're so cute to watch them. You know, because if you got a wet prep from somebody, a urethral discharge or a vaginal discharge, you can see them swimming around, just like we saw the proteus the other day swimming around. And I'll talk more about other ones later. There are these classifications depending on whether they're amoeboid, which we said has false feet, or whether they have cilia, or whether they have flagellate. They have these flagellum. You know, so they're kind of broken down in these categories this way, but we will talk about them and add them to our list as we go. And then we said helminths. Which kingdom do they belong to? They belong to the animal kingdom. They belong to the animal kingdom. So when we think about helminths, we're talking about tapeworms, flukes, and roundworms. Tapeworms, flukes, and roundworms. So anyway. The adults are often big enough to be seen with the naked eye. That is definitely true. And some worms, some of these tapeworms, can be many feet long, right? But the diagnostic part of these are typically going to be their eggs. And because their eggs are microscopic, that's what we're going to look at in the lab. And we've got a lot of eggs to look at for some of these worms. Many of these, and I know this sort of freaks people out to think that they would have these worms, many, especially ones that are really long, but many of them are going to be self-limiting. So you would actually, some of them don't live very long in your system, but some of them actually do and can live for quite a few years. But then eventually the adults would die off. The eggs that they're having are being passed in your stool. So the eggs are just passing through. And the adults might live with you for about 10 years, but then the adults might be gone too in some of these species. Yes? Well, there's a lot. It depends on which one you're talking about, but like the common tapeworms that's around here is by going barefoot in pastures and stuff, you know, and so you actually just are barefoot, and if you step anywhere where there was an egg, which animals and their species would be out there too, then it can penetrate through your skin, like the typical hookworm. It can penetrate in your skin, and it goes through your bloodstream to your lungs. And then it sort of hatches from there, and it crawls up your trachea, and you swallow it to your stomach. And then it attaches through this little scolex here. It's got little hooks on it. It attaches in your intestines, and there you go.
Well, I'm not going barefoot.
Well, that would be a real shame. Right? So, you know, you can get some of these from, not in this country anymore, but from eating contaminated meat. Again, why we want, you know, certain meats. How many of you are thinking about becoming a vegetarian at this point? But anyway, you can get them from that. Some of these liver flukes and whatnot, some of them are not endemic to this country. So Asian and some Asian waters, we have like schistosomiasis is a common one that soldiers come back and they have. So it just depends on which one you're talking about, but some of them are really common here. And some of them are actually in other animals. So because they're in other animals, they're zoonotic, so you can get them from that. But some are endemic to humans, like pinworm. And let's go ahead and put pinworm down. Pinworm is actually the name of the organism, and you can just put a little check maybe, or you can do it, but it won't be for Wednesday. I won't add any more to this for Wednesday, okay? But pinworms are so common, and they're endemic in the human population. How many of you heard of pinworms? And I don't know if anybody would want to admit it, but and there's nothing to be ashamed of for it, but bound to be that some of you had pinworms because they're very common. Pinworms are called, the name of the genus and species is Enterobius vermicularis. Enterobius vermicularis. Enterobius vermicularis are pinworms, what people call pinworms. And they are endemic to humans, and they are incredibly contagious. So what these adult worms look like are like little pieces of rice, maybe, so like little pieces of rice. And what will happen is, and it's very common, but anyway, what will happen is that the adult female at night, or it's really, we say at night because usually somebody's being still at night. When the person has gotten still and kind of horizontal, the adult female comes out of the anus and lays her eggs in the perianal area. And you can sometimes see the adult female looking like a little piece of rice moving. But even if you don't, what you can do is you can take what's called a Scotch tape rep, and you can take this clear tape and tap around the perianal region and then look at it under a microscope, and you can see the pinworm eggs, right? Because that's where she lays. Now, what that causes is for someone's anus to be incredibly itchy. So typically, if your anus is really itchy, what are you doing? You're scratching it. So I'm not trying to be funny. I mean, this is why they're so contagious. So you're scratching it, and especially if you're a child, and maybe even if you're an adult, and you're scratching it, and then you put your fingers in your mouth. What did you just do? What did you just do? You just re-inoculated yourself. And so it's really hard to get rid of pinworms because people are often re-inoculating themselves, right? They re-inoculate themselves and they don't get rid of it because of that. But also, as soon as you start walking, you get up and you start walking around, your cheeks are rubbing together. The eggs. I'm just trying to make you see how this is so contagious. The eggs are now in the sheets, and they're in the sheets, and they're in your clothes. And you go to your little elementary school, and you can't keep your hands off of each other in elementary school. And so, what are you doing? You're doing this to everybody, right? And so it's whatever. And even people who change sheets, they know because of the sheets. It only takes one egg to get into your mouth, your nose or mouth. It's going to get to your stomach, and then it's in your intestines, and then you have them.
How'd you say you get it?
So you get them from other people. This is totally endemic in the human population. So they're all being given around people. So you're not getting them from your pets, and you're not. What you just said? How did it start? Wait, Mama, that's like asking me how life began. I don't know. Hey guys, these are part of our community, And by the way, these are kind of nothing. Because these don't make you, other than the fact that you might get a little nickname of scratching your butt. These are not going to cause you, but it's really common. And so what happens is you have to treat the whole family. And if they don't treat the sheets, and if they don't treat the clothes at the same time, then what's going to happen? They're going to get it all again. And so it's very, very common, yes.
So do we need to do this for our test?
Not, did I say for test Wednesday? No, but you just, after that, because we've introduced it, so y'all be
She
fair
said
to
just
me.
write it down.
So just write it down, but not for Wednesday. What'd you say? I was going to say Dakota's never going to forget it though. The pinworm thing. It's very common. It's so contagious that you get the notes home from schools. How many of you had a classroom where you had the notes home from schools over that?
No.
It was always like.
It was always, okay, never pinworms. Never pinworms? Oh my gosh. Well, I guarantee you, your schools see it all the time. They do.
They never said the same.
So you can have tapeworms that are flat, and you can also have round worms. So just all kind. We'll talk about worms though. We'll talk about life cycles when we get there.
Please.
We'll talk about these life cycles. Okay. They lay a lot of eggs. So here's that one. And again, we'll talk about life cycles when we're closer to that. So how do y'all feel? All right, I don't want to really exhausted. Disgusted?
Yeah.
Do you remember in here, can you actually be in too clean of an environment?
Yes.
Do we actually need our immune systems being challenged by all these things that are very natural?
Yes.
Are we disgusted?
Yes.
No.
Yeah.
No.
Semi.
No.
So just like if anybody you see my scratching,
Scratching their butts. Is that what you're saying?
stay away. Just turn around and go the opposite way.
So anyway, guys, all right, so look, as a quick review, we went through the stuff in chapters, I think, what was that, chapters 1 through 4, but I didn't cover everything. But then I also brought out some other stuff though, right? So gave some definitions and whatever. I'm going to be I haven't even written it yet, which I guess y'all could tell just how I'm talking. But I'll get these questions together and let me tell you what I'm going to do. I'm going to go back and look at some of your study guides. I am. I'm going to look at study guides. I'm going to try to remember some things that we talked about. And I'm going to pull questions. Right?
How many questions?
I don't know.
I love.
Do you have it? She does. That's what you do as a study guide. Don't you do that? You make tests. So how long life's been on the planet? How long's life been on the planet? What GMOs are? Look at this. I need this. What does zoonosis referring to? Yeah, things that you can get back from animals, right? So zoonotic diseases, that's what they are. Look at these questions. Have we talked about all this?
Yes.
We get busy. MRSA. We talked about MRSA. Probiotics. What'd you say?
I have a lecture. I do invite the questions. I don't have to do that.
It's cool. Okay, so there's a lot of stuff. I should be able to come up with 50 questions, right? Multiple choice questions. So if I were y'all, I'd go back and look at people's study guides. I'd do that. That's great, Casey. That looks great. You want Casey's? So okay. So guys, today in lab, which we can go on in there. What I'd like to do is especially. No, I was just thinking I wouldn't have to, especially things that you have checked off on here. I actually would like for us to do a little bit of microscope work with some of the bacteria. So just to kind of be looking at those and differences. So today, basically, in lab, what I want you to do from bacteria to fungi, I want you, and you can do this as groups, or you can just look at them all on your own. But in like at a table setting, I have somebody get a really good example of bacteria. What kind of magnification do you need for bacteria? So you need 100x objective, which is what magnification? 1,000 times. So I get somebody doing that. Then I am going to have some protists and then some fungi. So I really want you to see the 3 kingdoms, right? At least these 3 kingdoms today, and looking at the magnification that the difference in these and just how different they look, okay? So that's some basic things I just want you to do today in the lab, and I'll get these slides out for you and be giving you an idea about which ones would be good ones to look at. These stains that are made of these organisms, some of the stains have faded, so please don't waste time on ones where the stains have faded. Please don't. You know, y'all had looked at the other day, some of you had good slides of flagellum, and some of you had pretty good slides of the capsules around little tiny bacteria, but some people had really faded slides, and so don't spend a lot of time on this faded. Go get something that you can actually see where the specimen was and look at it, and then y'all can just share and look at these. Today's lab is all about you getting an idea of the sizes, the difference between bacteria, protists, and fungi. There is not going to be a discussion board for today. You guys just look back at study guides. We're back at study guides. Look at your handout that you got. Do you have any questions from me before we move to lab? And can I have about 15 minutes or so? Is a genetically modified organism. What does that mean? What does that mean when I say a genetically modified organism? So it just means like they're crops. There's all kinds of things that we have actually genetically engineered, usually to make the crops more productive, to make them more resistant to like fungal diseases or other diseases, to increase the yields, right? Casey, are you with me on this? Or is that ringing any bells? Okay. You're the one that had it on your study guide. Okay.